All right, ladies and gentlemen, welcome back. We're looking at part 2 of protein synthesis process. As you guys saw this before, we have our different types of proteins and more importantly our molecular protein bouncing around there. So just as a reminder, when we were looking at setup here, I'm going to stay with red. We have our DNA molecule here, and it gets split apart, and then we have our RNA strand that's formed, and each one of these bases are being added in a complementary fashion, so U's with A's and G's with C's. Don't forget also we have our 3 types of RNA. This mRNA is what we're going to be using mostly today, and the rRNA, not the ribosomal, the transfer RNA. That's what we're going to be focusing on today. All right, so let's look at this here. So we know that our mRNA, once it's been processed, goes to the ribosome. And the whole point of this is in order to tell the amino acids that make up a protein which order they should be in. Because whatever order they're in will determine what the process of the protein or what the function of the protein is. So just as a quick little visual reminder, our DNA is changed to RNA in the nucleus. The mRNA comes out of the nucleus and goes to the ribosome, and that's where the protein is going to be made. And this is what we're focusing on in this video. So it's called translation. So it means, as in translating a language, going from one language to the next. It's the same thing. The language that we're translating is from RNA into a protein. So in order to do this, there's actually a code. And what it is, it's a 3-letter unit of mRNA. So when we have something like G, C, A, C, G, well, 3 letters will determine what the amino acids are. So when we look at something like this, these are all the different possible combinations of RNA and the amino acid that's going to be associated with them. As you can see, there's a lot of repeats. So even though there's 64 different combinations that these 3 or 4 letters can be arranged in, only 20 different amino acids are going to be formed. So there's a lot of repeats. And the reasons for this we'll go over later. This will be given to you in class. You do not have to memorize it. There's only one that you actually have to know by heart, and that's methionine. Here, that's the start codon. I kind of remember it as it's AUG, and the reason why I remember that as the start codon is August. End of August is kind of the beginning of the start of the year, the school, whatever. Okay, what's transfer RNA? Well, we talked about mRNA, and that's going from the DNA inside the nucleus to the ribosome. But what the transfer RNA does, it has a specific function, and it brings amino acids to the ribosome. So it actually is like a transport molecule. And for every one of the amino acids, there's a specific tRNA molecule. And they're very specific. And what happens is one end of the amino acid binds the tRNA. And what the other end does, it has something called the anticodon. So before we were talking about the mRNA has their bases, and we call 3 of them together a codon. So what happens is this tRNA comes and it matches this codon with its anticodon. Just like we were talking about with DNA and RNA before, they're complementary, so is the anticodon on codon. And we'll get into that in a second here. So this is just a typical tRNA molecule. Don't worry about the loops. So this is where an amino acid will bind, and whatever the amino acid depends is what the anticodon says. So if it's GCA, it'll be a specific amino acid. Okay, so let's look at the actual process. As you can see, there's 3 steps. The first is initiation, second is elongation, third is termination, and we'll go into those into a little bit more detail, but they're pretty straightforward in terms of their concept. The first part is it starts. It's the start of the translation. The second step is making the protein grow, and then the last part is stopping of the process. Okay, so let's look into it a little more detail here. You should have this handout, in order to follow along. So let's look at it. So here's the steps. You're going to want to write these ones down for sure. So initiation. So what's happening here is the AUG codon. So in this situation, it's right there. It's going to tell the complementary tRNA molecule to come bind. So it's the start codon. We call that methionine. And what happens is the anticodon on tRNA molecule binds to the codon of the mRNA. Okay? And what that does is it brings the perfect amino acid. Okay? So that's initiation. It's only the starting. Step 2 is elongation. So in elongation, multiple tRNAs are coming into the mix in order to line up with their anticodons. And what this does is it brings all these different amino acids into a specific area. So more tRNA molecules are binding. That just means joining with the mRNA to match it up. What this does is it brings specific amino acids, and I'll just put AA together. And then as you can see here, is that when these amino acids, this amino acid, when they come together, a peptide bond is joined, or is formed between them. So peptide bond joins the amino acids together to create a chain. And that chain is called a polypeptide. And that's just the precursor to a protein. So once that folds, it's a protein. Polypeptide. Okay? And the last step with our termination, and hopefully I have enough room here. Let's just go to the next page. All the termination is just like we had a start codon, we have a stop codon. So stop codon. In this situation, they use the UGA. So tRNA comes, it binds to it, but the tRNA for that codon doesn't have an amino acid attached. So there's no, just bring that out of room here. Over here. So no amino acid for a stop codon. All it does is just stop the process. Stops process. Okay, so you probably have to pause that just to write that all down. But that's the process of translation, turning the mRNA into something that can be coded to get the amino acids all in one spot. So pause that and write it down. So just in a summary, what do you need to know? Why translation occurs? We need to make that protein. Okay, that's the whole reason why, is to get it from mRNA into some sort of protein. We need to know that mRNA codons match with the amino acid, and that tRNA is what brings it there to match the anticodon. You need to know the 1-AUG methionine start codon, and then know the steps of translation. So we're going to be doing some activities more specifically with this next day, but make sure you write down any questions and have yourselves a wonderful night.